Good morning, students. Today, we learn about the last point from chapter 6, that is 6.10 DNA fingerprinting. This point is from chapter number 6, molecular basis of inheritance, which comes under unit 7, genetics and evolution. DNA fingerprinting. Introduction. It is the technology used for identification of individuals at genetic level, that is at DNA level. This technology was first developed by Alec Jeffreys in 1985. Here is the photograph of Alec Jeffreys, who first developed this technique. DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as a repetitive DNA. These repetitive DNA are separated from bulk genomic DNA at different peaks during density gradient centrifugation. The bulk DNA forms a major peak, and the other small peaks are referred to as satellite DNA. So when the DNA are exposed to density gradient centrifugation, you will find the major peaks and the minor peaks. That is, the major peaks, they will form the bulk of the DNA, and small peaks are called as a satellite DNA. So already we have studied from the Human Genome Project that 99.9% of the nucleotide sequences are same in all the people, and only 0.1 nucleotide sequences differs, and that major part 99.9, it is nothing but the bulk DNA major peak, and the small peaks, which is 0.1%, it is the satellite DNA. So when the DNA are exposed to density gradient centrifugation, you will find the two types of peak, major peak and the small peak. And major peak is nothing but the 99.9% of the DNA, which is same in all people. And small peaks are the 0.1%, which are differs. It is unique to every person. These sequences show high degree of polymorphism and form basis of DNA fingerprinting. The inheritable mutations is observed in a population at high frequency. It is referred as DNA polymorphism. The principle of DNA fingerprinting is based on matching of VNTRS, means variable number of tandem repeats of DNA collected at crime spot with the suspect person DNA. So here VNTR means variable number of tandem repeats. It is also called as a mini satellites that show very high degree of polymorphism. VNTRs are very specific to individual and differs from person to person. It shows some similarities between the family members. VNTRs of identical twins are same, hence it is not possible to identify individually in identical twins by DNA fingerprinting technology. So here all these points are coming under the introduction part. So here you have to remember that the DNA fingerprinting, which is also called as a DNA profiling, this technique was developed by Alec Jeffreys. Then it is a technique which helps the people in identifying a person at genetic level, that is at DNA level. Then each individual has a unique of DNA in genome, and certain DNA segments have DNA sequence which repeats many times. It is unique to person. It is a technique used to detecting and comparing specific nucleotide sequence in DNA of individual, and thereby identifying the similarities and differences in such sequences. It involves matching of a short piece of DNA of a victim with the DNA of the suspect. So there are two principles, DNA polymorphism and VNTR. DNA polymorphism means inheritable mutations observed in a population at high frequency is called as a DNA polymorphism, which is the basis of DNA fingerprinting. And VNTR, variable number of tandem repeats, that is, DNA sequences in some part of human genome are repeated many times, which is called as a VNTR, that is variable number tandem repeats. It is also called as a RFLP, restriction fragments length polymorphism. Our next point is the main steps present in the DNA fingerprinting. There are 6 steps. First one is the isolation of the DNA. Second is the digestion of DNA by restriction endonucleases. Third is a separation of DNA fragments by electrophoresis. Fourth one is a southern blotting technique, is transferring of separated DNA fragments to nylon sheath. Fifth one is hybridization using labeled VNTR probe. And the last step is autoradiography, that is comparison detection of hybridized DNA fragments. So these are the 6 steps which will give you idea regarding the process of DNA fingerprinting. The 6 steps are very important. 3 marks question may be asked just to mention the steps present in the DNA fingerprinting. So you have to remember in this order only, that is isolation of the DNA, digestion of the DNA, separation of the DNA, southern blotting technique, hybridization, and autoradiography. 5 marks question, if it is asked, you have to explain each step in brief. So we'll see each step in brief. What is mean by the isolation of the DNA? So isolation of the DNA, DNA is isolated from the sample of blood, cells, hair root cells, semen or bone collected at the crime spot. The DNA of suspect also collected and isolated separately. So here the DNA are isolated. How the DNA are isolated, that already we have studied in the biotechnology. When the process recombinant DNA technology, we have studied how the actual process takes place. Here under this, just we have to mention how the steps occur in brief. So DNA is collected from the crime spot and it is isolated. DNA is isolated. So our next point is digestion of DNA by restriction endonucleases. The isolated DNA is treated with restriction endonuclease to cut into numbers of fragments. So isolated DNA is further digested by restriction endonucleases, which are called as a molecular scissors in the biotechnology. So here we have this point also we have studied in biotechnology in detail. The next step is separation of DNA fragments by electrophoresis. The DNA fragments are separated according to their length on gel slab using gel electrophoresis. DNA strands on gel slab is treated with alkaline solution to split double strand into a single strand. So here, with the help of this electrophoresis, the DNA fragments are separated according to the length, and here you will find the double-stranded DNA are converted into the single strand. About the electrophoresis, we have studied in the biotechnology in detail. Next, the transferring blotting of separated DNA fragments to the nylon sheet. It is called as a southern blotting technique. Here, the single strand DNA is transferred to the nylon sheet and the technique is called southern blotting technique. Our next step is hybridization using labeled VNTR probe, variable number of tandem repeats, that is VNTR. The single-stranded DNA is hybridized with radioactive probe of VNTRS. The excess of the probes are washed off. Nylon sheet is X-ray photographed to get bands of VNTRS. So here in this process, the hybridization takes place. And the last step is detection of hybridized DNA fragments by autoradiography. Here, the bands of X-ray sheet is the DNA fingerprint. Comparing the DNA fingerprint of sample collected at crime spot with the suspect identifies the individually. So this is the brief explanation of the process of DNA fingerprinting with the help of 6 steps. Here you will find the diagrammatic representation steps involved in DNA fingerprinting technology. Here the first diagram, cell sample. From this cell sample, the DNA are extracted. Then cleavage of DNA by restriction enzymes, the fragments of the DNA with the restriction enzymes. Then separation of DNA fragments by electrophoresis. Here you will find according to the length, the DNA fragments are arranged. Then the transfer to the membrane nylon sheet, that is southern blotting technique. Then the binding of radioactive DNA probe to specific DNA fragments. Then comes the membrane washed free of the excess probe, and you will get X-ray film used to detect radioactive pattern. And when it is compared, you will get the results. That is, you have to compare the DNA fingerprint of the suspected person and the victims. So here the 6 steps are there. Here the diagrammatic representation, you will get an idea how the process takes place. Our next point is application of DNA fingerprinting technology. The first application is solving disputed parentage. Second is to identify criminals and rapists. Third is reuniting the lost children. The fourth is immigrant dispute. The fifth one is to establish the identity of dead bodies. And last, in the field of genetic biodiversity and evolutionary biology. So one by one, we'll see in brief. The first one is solving disputed parentage. DNA fingerprints of a child and the mother and the suspected father are compared. The bands present in the child's fingerprint must be accounted for those present in the mother's and father's fingerprint. First, the child's fingerprint is compared with that of the mother. Their bands are marked. The remaining in the child's fingerprints must be present in that of the child's father. So here, to solve the disputed parentage, the DNA fingerprint is very useful. The second is to identify criminals and rapists. DNA fingerprint is a technique used in forensic laboratories to identify criminals and rapists. The bands appearing in DNA fingerprints of a suspected criminal are compared with those obtained from the DNA collected from the crime site, and its bands are matched, then the confirms identity. The third one is reuniting the lost children. DNA fingerprinting helped in reuniting the lost children with their respective parents who were separated during war, violence, natural disaster, baby switch off over in hospital, etc. Next is immigrant dispute. More or less in every country, there is a problem of immigration. DNA fingerprinting used for setting up DNA database. It is helpful to establish whether they have crossed the borders or are the residents of that country. The next is to establish the identity of dead bodies. DNA fingerprinting technique is used to establish the identity of person killed in fire, plane crash, or any other accidents. So in this way, the DNA fingerprinting is very useful. And lastly, in the field of genetic biodiversity and evolutionary biology, the DNA fingerprinting is useful. So in this point, we have studied the 3 aspects. First one is the introduction. There you will get the idea regarding the DNA fingerprinting. what is the principle behind it. Second is the steps involved in DNA fingerprinting. And third one is application of the DNA fingerprinting. So this is the last point from this chapter. Thank you.